Today I'll be introducing you to my latest research in the Department of Preventive Medicine. We will call it Incorporating Metafeatures into Epigenetic Modeling of Human Age. My name is Daniel Rudd, and I am a first-year student in the Biostatistics program. I am working under the supervision of Dr. Sigmund for this project. So, we all know that our unique DNA can tell a lot about us. Now, is it possible to determine our age by looking at the conditions of our DNA? Our aim is to create a model that can predict human age based on epigenetic data of individuals. But, because our age predictions are based off of DNA signatures, it is in some sense a measure of one's biological age. Therefore, it can give us some idea about how one's biological age compares to their calendar age. Recent research has suggested associations between age and the state of the epigenome. The particular epigenetic marker we will investigate is called DNA methylation. DNA methylation is a process by which a methyl group is added to a cytosine nucleotide of a DNA strand. DNA methylation is of interest because it has been linked to gene expression and suppression. It influences many processes including cell differentiation, genetic mutation, disease progression, and many more. In particular, we examine DNA methylation at locations called CPG sites. Now, what are CPG sites? Well, we know that DNA is comprised of 4 nucleotides: adenine, thymine, cytosine, and guanine. CPG sites are the locations on the DNA strand where a cytosine is followed by a guanine, bonded together with a phosphate molecule. CPG methylation sites are measured using the Illumina Infinium Human Methylation 450 B-chip assay. Because a single sample of blood or tissue contains many cells, we can measure methylation by looking at the proportion of times a site is methylated across all cells in the sample. More specifically, CPG locations across blood cells in a single sample are used to generate our methylation proportion data per individual. To build a predictive model of age, we will first explore a dataset which contains thousands of CPG methylation proportions across about 650 individuals. To get a small preview of the dataset, we can see that the methylation proportions at different CPG sites are recorded as the columns of the table, while the rows pertain to the individuals sampled in the dataset. Finally, we will incorporate novel metafeatures into our predictive model. Our goal is to find which CPG locations are the most important for predicting age, while adding metafeatures to help build a stronger model. These metafeatures are prior pieces of information about CPG sites. They can include gene affiliation, location of CPG site within genes, if a CPG is located near an enhancer site, and many more. Garrett Weaver and Dr. Lewinger have created an R package called XRNet, which allows the incorporation of metafeatures for our modeling purposes. We hope that the incorporation of metafeatures will help us understand underlying biological mechanisms. Similar research has been produced by Dr. Steve Horvath and Dr. Gregory Hannon. Our novelty comes in the addition of metafeatures to improve on existing models. Because we are interested in creating a generalized linear model, while imposing the necessity of feature selection, the optimal choice of model is a regularized regression model. This will help us choose which CPG sites are the most important for predicting age. In our preliminary analysis, we explored a subset of 10,000 CPG sites from the dataset. Our feature selection found that around 160 CPG sites were important in estimating predicted age. We plan to continue on with the analysis, incorporating metafeatures to see if our model prediction improves. Thank you very much for listening.